established, and this basically is where we will start in just a moment. But we recall that with the cranial and caudal folding and the lateral folding, that coelomic space became incorporated into the embryo, and then toward the midline or medial to that coelomic space, we ended up with different types of mesoderm, the paraxial, the intermediate, and the lateral, within which, of course, was these coeloms. These coeloms. The paraxial, obviously just parallel to the center of the body, just lateral on each side, are going to differentiate into two parts. The sclerotomes, these will form the vertebra and the ribs, and the dermomyotomes that are going to contribute to the skin, the muscle, they will form the muscle, the musculature, and contribute to the formation, not really, I guess, of the skin, but really of the connective tissue underlying the epidermis. So the dermis and hypodermis basically will be the components of this. So contribution to the integumentary system is a term that I would prefer because the skin is epidermis and dermis and does not include the hypodermis, but here we are thinking of the integumentary system that includes the skin, epidermis, dermis, and of course the hypodermis. And mostly the vasculature and the connective tissue, as we know, originates from the mesoderm. The neural innervation is already there, motor efferents from the spinal cord, and other efferents and afferents, the sensory coming from the dorsal root ganglia, the spinal ganglia, of course, originating from neural crest, and the postganglionic sympathetic fibers that we would find here originating also from neural crest. So all of these components are there. The same for the musculature, the entire neurovascular bundle, and let's not leave out the lymphatic vessels for all of these structures as well. We start then with the illustration, or figure 15.1, and we'll see that it will illustrate the formation and the early differentiation of the somites. We start in this panel A with the dorsal view of the pre-somite embryo, so there's no somite present yet, about 18 days. Then we'll go to a transverse section as shown here in A, the level of section for panel B. And we'll see the paraxial mesoderm that will give rise to the somites. Then we'll look at a transverse section of the embryo about 22 days, so about 4 days later, where we'll see the early somites beginning to appear. We'll look at the neural folds, our old friend for orientation, neural fold, neural tube. And at this stage, of course, it is beginning to fuse to form that neural tube. Then we'll look at another transverse section about 2 days later, 24. We'll see the lateral foldings of the embryo in this transverse plane and see the differentiation of the paraxial mesoderm into the dermomyotome region. And of course that gives rise to the dermatome and myotome, just further differentiation of the dermomyotome to separate into that which will contribute to the skin, and of course that which will contribute to the skeletal musculature formation. And of course the scleroderm is going to be present as well, and we'll see that all three are present when we look at the 26th day in E, dermatome, myotome, and sclerotome. Again, this is the pre-somite, the day 18 that we saw, and we recognize it, these coelomic spaces forming in the lateral mesoderm, most medially located in this area, obviously just to each side of the notochord that has triggered the development of the neural plate to differentiate into the nervous system, as we know, will be the paraxial mesoderm, and that's our focus for today. And then in between the lateral and the paraxial, of course, is the intermediate. As I stated earlier, the overlying epithelial layer, ectoderm, and the underlying endoderm with the differentiating mesodermal layers between those two, and paraxial, intermediate, and lateral is what we see here. This was 4 days later, if I recall, the 22 day. So the neural folds are quite accentuated, and there's the early signs of the beginning of the fusion of the neural folds. We know that very soon after this, the neural crest cells are going to originate from that junction of the surface ectoderm and the neural fold. We see some changes in the paraxial mesoderm. It is now given rise to a collection of cells in this area, giving on the surface that bump that's characteristic for the somite. So it is now referred to as the somite. The intermediate mesoderm appears smaller relative to the lateral that now has a more extensive intraembryonic coelom. We now go to a more illustrative appearance. I think this was the day we went to 24. Excuse me. 18 to 22, and now we're at 24, then we'll go to 26. So this is the day 24, 2 days later. Lateral folds are very obvious. We orient neural tube, dorsal, most structure that we've identified throughout, and we see the two dorsal aorta, and this structure, of course, is the notochord. On either side, right and left, if it's appropriate for this view, we can see the dermatome closest to the epithelial cells on the dorsal aspect, the ectoderm, and a sclerotome, obviously more medioventrally located. Dermomyotome, I should restate that. Now we proceed to a couple more days. Advanced in this stage of development, we see that the lateral folds have reached the ventral midline. Of course, they do not fuse completely where the primordial gut tube, basically the umbilical cord, the yolk stalk attachment, or midgut primordium, is present. And of course, this should represent the amniotic sac here, because we know ultimately it completely encases the embryo. The intraembryonic coelom has now become those spaces, the two balloons that we've talked about coming around, like the butterfly wings, capturing these spaces on each side with basically the gut tube in the center, forming the pericardio-peritoneal canal, essentially, right? And this will be, because we see the midgut primordium here, we know that this will become the peritoneum. Peritoneal cavity with the dorsal mesentery here, and other than this location, the ventral mesentery on these structures. Back then to the dorsal orientation, the neural tube. We see the neural crest cells had begun to develop and now are differentiating into the spinal ganglia. At this time as well, we know that the neurons in the ventral spinal cord or neural tube are beginning to send out their axons, so the neuronal fiber connections to these developing structures is already being established. So as we begin to form the dermatome, the sensory and visceral fibers are already beginning to move into that area to establish their connections with their target cells, target organs. There's a thin layer in between, the myotome, and then the sclerotome. Our focus today then is, within all of this, to discuss the development of the bone. So we'll work on the skeletal system development today. Bone is a connective tissue, therefore it is of mesodermal origin, except for the neural innervation of it. But everything else will be of mesodermal origin. This is with the exception, of course, of the specialized mesoderm in the pharyngeal arch areas, We recall that the specialized mesenchyme there was produced by the neural crest cells. But just the same, it is ultimately mesenchyme, whether it's of mesodermal origin or neural crest origin, depending upon where we are in the body. The flat bones, our example, the skull, will develop by a process that's called the intramembranous ossification. So obviously we're going to lay down some type of fibrous membrane structure. Cells will have migrated into the core, if you will, of this membrane and begin to differentiate from mesenchymal cells into bone-forming cells. And the bone-forming cells include both the those that will lay down the bone and those that will take up the bone for remolding. And let's just wait for the specifics on the origin of these, because they do differ respectively from each other. So the osteoblasts, as we know, are those that will develop the matrix, lay down the first hydroxyapatite, the calcified deposits called osteoid. The calcium salts become trapped in that osteoid. This is the osteoid is the extracellular matrix that contains everything essential to producing from its contents the calcium salts. And of course the cells will become embedded within this, and we'll look at this differentiation of the primordial bone-forming cells all the way from osteoblast to osteocyte. There is remodeling that occurs, and this is, of course, throughout life, but also through the embryonic and fetal development, and it is by the action, coordinated action of the osteoblasts and the osteoclasts. So let's look at this slide to investigate or study the developmental lineage and final appearance of these two types of cells, these antagonistic types of functions, laying down bone on the left and removing bone, especially the calcium deposits on the right-hand side. So early from the mesenchymal cells, there will be an osteogenic cell that develops into the osteoblast. The osteoblast forms the bone matrix, and as soon as it is surrounded completely by the ossified bone, of course it does change shape. It sends out these protoplasmic extensions. In adult bone, we know that these reside within the lacunae, the canaliculi. The main part of the cell body resides within the lacunae. So in ground bone, we can see the dust-filled spaces where these structures had resided. So this is the lineage of the osteocyte from a mesenchymal cell. The lineage for an osteoclast actually is from a white blood cell. And goodness gracious, at the moment, I can't remember which one. The one that typically will develop into a mast cell, a macrophage as well. Monocyte? Yes, the monocyte will come from the vasculature through the endothelial cell wall, cells lining the walls of the capillaries, and migrate into these areas and be stimulated to differentiate into the bone-removing cells, the osteoclasts. So still, obviously, blood, including the white blood cells, are categorized as connective tissue, and, of course, still of mesenchymal origin, mesodermal origin, but the osteoclasts do not develop from locally generated cells. There are local stimuli to bring about the differentiation of these cells that would otherwise become macrophage to become associated within bone as osteoclasts. Here then in figure 15.4, we see a light micrograph of the example of intramembranous ossification. The dark pinkish colored structures are the osteoid, basically, that remains. This very likely was decalcified. We're seeing the pink staining predominantly of the amorphous gelatin type of extracellular matrix molecules, and as well any type of fibrous molecules that are there, such as collagen. We see shrunken dark blue structures, if my mouse would stop sticking. Inside these little spaces, the space would be the lacunae that I had mentioned, this lake within the bone-forming area. So these would be osteocytes. The shrunken up, shriveled little dark blue spots would be the osteocytes. They've shrunken and pulled away from the margins of their lacunae. Then out here, of course, this is the non-ossified space filled with the mesenchyme of cells differentiating into fibroblasts, the vasculature, the neural innervation would be in this area already. We can see one blood vessel right here, the red blood cells captured within it, another one there, and another one here, and another one here. Typically, this characteristic appearance is that of veins. Not necessarily definitive, but that's what we would predict. So we see many mesenchymal cells, these blue squash dots that are differentiating into the various cell types there. And then the cells that are lining these trabeculae, as they're called, the primitive, where did it go? Okay, yeah, the trabeculae. So these, lining all these trabeculae will be cells of two types, osteoblasts and osteoclasts. The osteoclasts will always stay on the margins of the trabeculae, and the osteoblasts ultimately will lay down enough matrix to surround themselves so that they are differentiated thereby into the osteocyte. So the primitive osteons or canals are evident in places like this where these large blood vessels are trapped in the core of them. So all the characteristic features of mature bone are reflected with the structure that we see here. We can now go on to this series of illustrations to follow the development of flat bone, for example, the skull, to the point that we just looked at in the previous actual histological sample. We are then considering a flat bone of the skull where these events are beginning to occur, and an ossification center will begin to form where some of the mesenchymal cells will have differentiated into osteoblasts and begin to lay down the osteoid, here shown in the white, referred to as the center of ossification. Nearby mesenchymal cells would have differentiated into blood vessels much earlier on, obviously, but there still will be ongoing angiogenesis. It hasn't ended yet, of course. And then in the surrounding mesenchyme, we see mesenchymal cells in this area, not quite yet committed to one cell type or another. Many of these, of course, will become fibroblasts that maintain the connective tissue surrounding the bone. And we'll talk about the development of that periosteum in a moment. So these initially trapped cells, although they're supposed to be there, so they're not really trapped, but those that become embedded in the center of ossification will go on to lay down the within the osteoid the calcification byproduct. Here they're showing the canaliculus, where these cytoplasmic processes reside. So the lacunae would be where the main cell body resides. So the characteristic feature of a bone-forming cell completely surrounded by or encased by ossified bone is now an osteocyte. On the surface of this early structure is the osteoblasts. Some of these might be the primordia of the osteoclasts that have come from the vasculature and are sitting here as well. But by far, the majority of these are going to be the osteoblasts, beginning to lay down bone on the surface, still mitotically active. That's why they have that term blast. They are capable of mitosis, and one daughter cell goes on to lay down the matrix, ossify it, and surround itself and become an osteocyte. Here is some evidence of both new angiogenesis as well as increase in the dimensions, the overall size of this developing bone trabeculae, so that it now encroaches upon some vasculature. Here just the artery is shown, and of course it will surround it and lay down bone around it, forming the osteon, the cylindrical columnar hole, if you will, through which the neurovascular bundle, it's not just the artery, it's the full neurovascular bundle, will pass to the innermost portions of the bone. So here we can note that a canaliculus is terminating here near the osteon, the early osteon, and of course the osteon has within it interstitial fluid exchanged with the capillary beds in there as well. And it is that interstitial fluid in the osteon that these protoplasmic or cytoplasmic processes within the canaliculi make everything happen. They receive the stimuli, hormonal stimuli, either to lay down bone or to decrease the rate at which they lay down the bone, and for all the nutrients and waste exchange deep within the bone. So these osteocytes are busy communicating with each other. There isn't much interstitial space outside of them, but that interstitial fluid is crucial, as you know, because you've got all the events for maintenance of the bone, and you've got all of the exchange of nutrients and waste and so on and so forth. And then we see still the osteoblasts on the surface. We also note that the surrounding mesenchyme is condensing, beginning to contribute to the connective tissue layer that surrounds the outside here of flat bone. Ultimately will become the periosteum, this layer with the osteoblasts and this layer of condensed mesenchyme. Ultimately, they will become the two layers of the periosteum, the osteogenic layer, the innermost portion. Obviously, you want that just adjacent to the bone. And the fibrogenic layer, forming that fibrous capsule, if you will, that surrounds the bone. A more mature appearance is shown here with the spongy bone in the center that has two plates of compact bone on either side of it. Remember, we're discussing flat bones here. And the periosteum, the two layers being identified, the fibrogenic outermost and the osteogenic innermost. And then what isn't being shown here is in both the compact bone and the trabeculae of the spongy bone would be the osteocytes and their lacunae with their protoplasmic processes and in the canaliculi, and the osteons that contain the neurovascular bundles. So this osteogenic layer of the periosteum has the capability of mitosis and producing daughter cells and a reserve cell. The daughter cell can go on to differentiate into an osteocyte and lay down new bone. In embryo, fetus, and neonate on through pubescence, obviously, there will be new bone laid down. But even throughout our lives, Bone, when we're less active, it'll get thinner. When we're more active or weight bearing and things like that, then it can add bone. So it is still a dynamic layer, that osteogenic layer. And all of the signaling that tells it to do this or that, I'm sure, is very complex. Most other bones, therefore, the long bones, for example, are going to develop by endochondral ossification that begins at about week 5. So there will be a primary ossification center, and this will be in the center of a long bone. So picture the bone in your arm from the shoulder to the elbow. The humerus, in the very center of it, would be the primary ossification center. And then that's called the diaphysis, or the shaft of the bone. And then on either end, sitting on top of the shaft, the epiphysis on each end, or epiphyses, it would be plural, where we'll see the secondary ossification centers. So this first one, as we will see, also corresponds to very closely to the area of entry of the nutrient artery, the main arterial supply to the diaphysis of long bones. So there's obviously an association between angiogenesis and osteogenesis. The process continues from that dead center in the shaft toward the epiphyses, so from center to the ends, toward each end, where ultimately it's going to meet the epiphyseal portions and join and unite with the effort of forming bone there. And we know that that is at the epiphyses, where the epiphyseal plate or the growth plate is going to reside for the lengthening of the bone once the shaft of the bone is formed. And again, it's a coordinated effort of osteoblasts and osteoclasts. So throughout, there's formation and reformation of the bone, and it requires that coordinated interaction of osteoblasts and osteoclasts. And as we had stated, there's remodeling throughout life. Excuse me. Here in this illustration, panel A of figure 15.5, it's a schematic of the longitudinal section through a long bone, as we had mentioned. We're seeing the intercartilaginous or endochondral ossification of the developing long bone. So a simple illustration here on the left. And then we magnify or enlarge this area here. First, let's orient to the outer portion, the mesenchymal layer with the mesenchymal cells. It hasn't quite condensed yet, but it will, and contribute again to the fibrogenic layer of the periosteum. And then chondroblasts are formed from the mesenchymal cells inside. And once these chondroblasts surround themselves with cartilage, chondro, the prefix chondro means cartilage, so endochondral formation. We're going to lay down cartilage. It's a hyaline cartilage, and then cells will come in forming osteoblasts and osteoclasts, and then replace that with osteoid and calcification. So the perichondrium is beginning to form at its margin as well, and with a lot of mitosis occurring here and differentiation of cells as they surround themselves with matrix. When the periosteal capillary, or the nutrient artery as I had called it before, we'll see it identified as that a bit later, invades this area, the osteogenic events are altered or triggered by this angiogenesis. So the primary ossification center is going to be here at the core, but as the vasculature invades, there will be the junction with the epiphyseal plate, and we see now the chondrocytes are aligned here at this junction of diaphysis and epiphysis, and the hyaline will begin to be calcified. Osteoblasts and osteocytes will take over. but there will still be at the distalmost ends of the bones uncalcified hyaline cartilage. This becomes in part the growth plate, the epiphyseal plate that persists through puberty. But of course, on the articulating surfaces, there will be some of this hyaline cartilage that will persist as the articular cartilage. So there isn't a complete replacement of all of the cartilage even throughout adulthood, although we might wear and tear our joints and destroy it, and it can't be replaced. It was supposed to be there. The periosteum becomes well developed, again with the fibrogenic layer, the outermost, and then the osteogenic layer, the innermost. And we see the subperiosteal bone forming the bone collar of the diaphysis. This continues throughout the development, of course, and the secondary, with the angiogenic events in the epiphyses, the distalmost ends of the bone, there will be the secondary ossification centers that will form, and the cartilage in that area as well will be replaced via osteoblasts and osteoclasts, the osteoblasts ultimately differentiating into osteocytes to form the typical type of bone that we recognize at the ends of the shafts of long bones. So it's going to be the spongy bone with the dense bone outermost portion. We know that the osteons in the shaft of the bone become those haversian canals, so they become long cylindrical structures. And this is because of the way that the vasculature comes in. They're not showing the branches that end up in the osteons, but there will be these branches going in, and they just form these long ascending and descending branches, and the bone forms around them. So we end up with this mature-looking bone, the nutrient artery, as I said, coming in at the mid-diaphyseal point. It had triggered the primary ossification center. We see the bony collar, the dense bone, and then some evidence of the trabeculae. And of course, we know that the mesenchyme within this space in the spongy bone becomes the bone marrow. So we don't want to leave out the bone marrow. And this, of course, reminds us that all the blood cell formation, all the hematopoietic tissue for both red blood cells, the erythropoietic cells, and the white blood cells, the leukopoietic cells, and the platelets, the megakaryocyte-forming cells, poietic cells, all differentiate from mesenchyme that was in this area. So we don't want to forget that. And then ultimately just becomes fat cells in most of our long bones. Here in the lower portion, we're seeing a bit more of this vasculature, even though they're showing it within the bone marrow cavity, this appearance of ascending and descending vasculature, and it would be neurovascular bundles. And you have to imagine these out here in the dense bone too, within the haversian canal systems. I would like them to have shown some of that as well, but it probably was considered to be distracting from their main point of reminding us that the angiogenesis is very tightly connected and coordinated with the osteogenesis in all of the bones, as we've seen. And the angiogenesis brings the capillary beds through which pass the monocytes that differentiate into the osteoclasts. And very likely some type of signaling targeting many of the cells in this area as well. So what is meant by the rearrangement of the sclerotomes and development of the vertebral column? So we're going to transition, I guess, on Monday, since our time is up, to a discussion of the vertebral column and we'll start with this slide at that time. Appreciate your time and attention, and hope you all have a good weekend.